So I'm briefly going to go over this laboratory kit with some of the experiments that you're going to do. Now you have a bunch of things that you're going to do that are worksheets, some online, some things you're going to be doing at home with materials that you have at home. And there's a lot of data analysis labs, so data sets that I'm giving you, scientific papers that you're reading and going through, and so forth. That's like part of the class, one chunk of it. Another chunk of it, you're going to be collecting your own data. So you're going to have to be doing experiments or field data collection. And so that's what's kind of in this kit, is some of the things that you need to do some of that other work in the class, okay? So I'm just going to go over this very quickly, what sort of things you have and like where you would be using them. All right, so one of the first labs in here, it's an experiment that you have to set up. It's called Darwin Seeds. So now with each of these, this is not the explanation on how to do each of the experiments. This is just a quick overview of all of them. There'll be specific explanations on the Darwin Seed experiment. So for Darwin Seeds, what that's going to entail is you're going to create salt water, and you're going to be using these little cups that are in the kit. They'll be used for a number of different things in the lab. So one of them will be just to store some liquid. You can use any other cup for this as well. But they're just provided for you. You're going to be using them to plant organisms. So you're going to have some soil. You'll be putting soil on these to grow up like a little plant pot to grow up some plants. So that's kind of another use of these. That's why you have quite a bunch of them. About 20 are in this kit. So for the Darwin seed experiment, you're going to note you have different seeds. So in this tube is another tube, and in this tube there's another tube, and there are different species of seeds. And they should be written on it to what type of seed they are. So there are sunflower seeds and beans, and there are radish right in here. There's a lot of radish. So radish are going to be used for multiple experiments because there's so many. So make sure you keep track of them. So when you do the Darwin seed experiment, you're going to be taking some of these seeds and putting them into salt water, and other seeds are not going to get exposed to salt water. And then after they've been in the salt water for a while, you're going to determine how well they can germinate, like can the seeds develop into little plants or not. So are they affected by saltwater exposure or not, is kind of the idea behind that experiment. So that's kind of this one here. You should have what you need in the kit, but again, if you didn't for some reason, you need plant seeds. So something you can pick up on your own. You're going to need salt water, so just salt and water. I'm going to give you with the experiment exactly how much you need to mix. Then you're going to have germination conditions. So that's going to be just using the radish seeds, and you're going to have a whole bunch of little plastic bags. So with the little plastic bags, you're going to be putting a little piece of paper towel in them, just getting it wet. And then you're going to be putting some seeds into them. Some of them you won't be getting wet, so there'll be different kind of treatments that you're going to have, and you're going to determine under what conditions will the seeds germinate best. For this, you're only going to use one species of seed, just the radish. If for some reason you don't have the radish, if it wasn't in your kit or something, you picked it up really early and missed it, then you just any other plant seed you can do that with. So you can just pick one up for like $1 at a store. Then you're going to look at plant growth conditions. So after you have germination, then you're going to be using these little cups as little planters, and the soil, you should have either one big bag of soil or 2 small bags of soil. You'll be putting the soil in here. The germinated little seedling, you'll then be planting, and then you're going to set up an experiment of your own where you're going to decide which conditions, different types of light conditions, different types of water conditions, change the pH of the soil. You can do different things. So there's going to be a variety of options for you, and then you're going to see how that affects the plant growth, and you'll be measuring the growth. So there is a ruler, in here as well. You'll be kind of measuring the growth with that. So that's given to you. So you got the Darwin plant experiment, the seed germination experiment, the growth of a plant experiment. There's Artemia hatching. So in one of these, it's not plant seeds, they're little Artemia cysts. Okay, so there's going to be an experiment where you're going to be hatching out these little shrimp. And same thing, you're going to be getting salt water, though at different concentrations. So from fresh water to oceanic salt and higher concentrations, and putting them into it. But you'll be using these bigger tubes, for that. So you'll be putting liquid in here with those. When you're going to look at them and count them, you'll take some out with a little pipette, put it in this little dish on a surface where you can actually see the unhatched cysts and then you can see the little shrimp swimming around, and you can kind of count them with that. You're going to have some natural history studies where you're going to be collecting data, and for those you're going to need a balance. So there's a digital balance for you to weigh things, and so you're going to have to weigh out. You can use them probably for the salt, making the salt solutions and all. When I give you the instructions, I'll give you how much to weigh, how much salt to mix with water to make the right concentration. So you'll be using it for that, and so because of that, you have a little weigh boat to go with it. But for the other experiments, you'll be weighing like a pine cone or an acorn or a leaf, different things you're going to choose, but I'm going to be going over those specifically. You're going to pick a species, you're going to collect samples from that species, you're going to be weighing like 100 of them, and graphing them in a couple different ways. Then you're going to be collecting another set of samples from a different tree in a different location or a different flower or whatever it is that you're studying, because it can be partly your choice, and you're going to do the same thing over again, and you're going to compare them. So it's going to be two separate studies. There's natural history study 1 and 2. One is the first part of it, the second part is kind of repeating it with a different organism and then comparing it and combining the two together. So you definitely need a balance for that. You'll typically need the ruler for that. To do it, you might need, there's a little bit of string in here, which is used to measure diameter of things, so you can use that to go wrap around and then get the actual measurements that way. There are going to be population density ones, which is what you'll use this string for as well. The string is 2 meters in length, and so what you'll be able to do is tie the ends together of your string. I'll actually do it for this one. And then it'll create a little loop, but the idea is you're going to want to make actually more of like a square with it. So it'll be a 1/2 meter square, right, when you actually make a square with this on the ground, and I'll be going over that, but there's a population survey. You'll be putting this on the ground somewhere outside, and then you'd be counting things, say like a specific type of flower or plant or something in this, and then be repeating it over and over and over again multiple times, so you're going to have that data collection. You can make an apparatus out of a PVC pipe or wood or something like that if you want. It'd be much easier to work with, but I just couldn't include that with the kits, so this is approximation, so you can do the same study and it's much easier to pack this up. So you're going to have study like that you're going to do as well, and then you're going to have some scavenger hunt studies, which are essentially I'm going to be giving you lists of organisms or structures of organisms to find, and all you're really going to need is a camera to take the pictures of them, and then you're going to have to be labeling them and sending them. So some of them will be very specific structures of organisms, and others will be diversity, different types of organisms that you're going to be doing. So you have to be going outside to do that. But for the kit for the experiments, you're going to have some different seed germination, shrimp hatching, plant growth experiments, and then for several different data collection experiments, you need the balance. And that's pretty much it.